This is Sciographies, an introduction to the people who make science happen. I'm your host, David Barclay. I'm an oceanographer with the Faculty of Science here at Dalhousie University. And on Sciographies, I interview different types of scientists about what shaped their interests, their career path, and how they get their research ideas. Thanks for joining us. In this episode, we talk to Dr. Erin Bertrand. She's a marine biogeochemist looking at the relationship between microbes and ocean metabolism. To do this, she travels to challenging environments like the Antarctic to collect samples of ocean water. Dr. Bertrand talks to us about the overwhelming decision to choose a direction in university, the satisfaction of doing physically demanding work, both as a researcher and as a trail runner, studying how tiny microscopic changes have cascading effects in entire ecosystems, and her commitment to creating an inclusive lab environment. My favorite Erin Bertrand story is we had a flight together at 11 a.m. And I'm like 11 a.m. That's a very reasonable time for a flight, but actually you have to be ready to go like at 9. It's actually kind of a morning thing, right? So we get to the plane. I was like, I had rushed a little bit, and Erin was like, "Oh, my legs are so sore. I ran a ultra trail marathon this morning before coming here." So, yeah. Anyways, that's my perspective on this interview. Yeah. So I think in that context is where I'd like to start.
Okay.
Yes. You've always been a highly ambitious, productive child. Even.
Well, yeah, when I was a kid, I loved school. School was so much fun. I loved learning everything. I couldn't decide what was the most interesting subject, and it drove my brothers crazy. I have brothers who are twins. They're 16 months younger than me. They have many things going for them, but loving school was not one of them. So to follow on from me in our tiny school in rural Connecticut, the expectations for them were that they would be like me and love school. And
yeah, they're
not
like,
how
all the teachers were like,
goes.
the Bertrand boys, they'll be such a treat.
Yeah.
When you say you couldn't choose an area, is this sort of like a Rushmore, you were president of the French club, the math club, and stuff like that?
Yeah, perhaps. But I tended to stick with things I wasn't particularly good at either. So I would, keep playing the trumpet in band, even though I wasn't very good at it. But no one else was very good at it, so they gave me solos, and then I would fail to play.
This is the interesting psychology here. To read between the lines, you probably were a very good trumpet player. So when you say rural Connecticut school, you talking, how many rooms we talking?
Oh, it wasn't that small. There were 500 kids in the school, I think. So it wasn't that
reasonable
small.
size.
Yeah, it was a reasonable.
And that's a high school or all 8 to 12?
8 to 12.
Okay,
yeah.
so right. Yeah, I'm trying to envision you in your 8 to 12 school.
Yeah, I was one of those kids that I did everything. I played sports. I was in the band. I enjoyed all of that. I loved my science classes. I loved my English classes. That was kind of crippling in a way, because then I ended up in undergrad, and I felt like everything is amazing, and this is all way more exciting than I could have even imagined it would have been when I left rural Connecticut. And I just wanted to do everything. I couldn't decide. So that started early for me, which was fine.
So where did you end up in Maine? You were at Bates College?
I was at Bates
right?
College, so
and
small liberal arts
that's sort
college.
of just outside of Portland, right?
It's north of Portland in Lewiston.
Okay.
And Lewiston's a more interesting place. So it's heavily influenced by past and recent immigrant waves. So when I was at Bates, the Somali population grew really, really quickly when I was there. And so that brought with it a lot of the realities of the problems of the early 2000s with the racism that they were encountering on a daily basis. And so that was something that really struck me, and was completely different to what I'd grown up with in rural Connecticut, where there's economic diversity, but no other real kind of diversity.
Sure.
There.
So you end up at college. Are you in the Faculty of Science or Arts, or it's a liberal
arts school?
Right?
Yeah, and that's what attracted me to it, because I couldn't decide what I wanted to do. But I went in. It was the Bush years. I thought that's as bad as it was going to get. So I was going to go into political science at first and try to do something about all these problems that I was becoming aware of and worrying about a lot. So environmental degradation was something that I stressed out about all the time, and any number of societal problems. So once I got myself out of that little town, I could see more clearly all these problems. And that's sort of all I started to see when I first went to undergrad.
Yeah.
And because I was interested in everything, I didn't know what to do and how to help and where to direct myself. And I found that to be pretty crippling.
Yeah.
But I took all sorts of poli-sci classes and I loved them, but it was emotionally really, really draining for me and I felt like every assignment I was sort of pouring my heart and soul into, because it was hard to think about all these problems and not be able to solve them immediately. So I would do this mundane paper with everything that I had.
Yeah.
And that couldn't continue, I decided.
Yes.
So that's part of why I chose to move into a path of natural sciences.
Were you out there like rocking against Bush and other things?
Oh, yeah.
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 I mean, obviously it must be hard if you're living in Lewiston to go out and protest or...
Yeah, I mean, so I mentioned the Somali immigrant community. There was a lot of attention on Lewiston at that time.
Okay.
People were coming there. The Bush twins came to rally the Republicans in Maine there. And there was a lot of really vile white supremacy that was coming to town. And so I was out a lot organizing against those visits to town. I think this is part of my move to natural and biophysical sciences was to give myself dedicated things to do in the lab so that I didn't get emotionally derailed by what was going on around me. And it felt like I could make an impact there and do something that mattered, that was important, without losing myself. Yeah.
So in terms of worry, I mean, obviously, say you had become a political scientist or whatever, in working in that realm.
Yeah.
Things are a lot more difficult now than they were back then, is the general feeling, I would say. Mm-hmm. But also in the natural sciences. What do you think's gotten worse?
Well, so now, I mean, there are more demands on us as natural science researchers to think about how our work impacts communities and humans directly, and I think all of that is really good. But it's starting to bring me right back into that place where I made decisions to protect my psychology. And
exactly.
Now I've got to do that all over again and make sure I set myself up to not get buried.
Yeah.
Because in that worry.
It's maybe not totally directly, but in general, your research has links to...
Yeah, it has links to climate change.
Climate change and the big questions of our day.
And all research is embedded in the system of white supremacy. Mm-hmm. All of these things are things I think about daily now. And when I was in undergrad, I had the impression that natural sciences, biophysical sciences are empirical and they are, you're just trying to understand the world.
Right.
But obviously now we understand that isn't true, that there are all these lenses that we bring to our work as natural scientists. Mm-hmm. And so what I had taken as a way to protect myself, now I'm thinking about all those things again, but I have a lot more skills to think about them. And I think that's only good. And in some ways that's been a really satisfying change in what my day-to-day work is like. Mm-hmm. So I can re-engage with those questions that I always knew were ultimately important without letting it cripple me.
Sure.
So, yeah.
And do you feel in that regard like your role as you become more senior and more established allows you to spend more time on those questions and those perspectives than you would have been able to earlier?
Exactly. Mm-hmm. Mm-hmm. Or I feel like I'm now in a position where I must do that.
Right.
Yes. Yeah. And that's a good thing, I think. Yeah. Obviously,
this is something I think we all deal with as we're going on in our career. Yeah, how to be a bit of a leader, how to bring some thoughts that we're not necessarily trained in, but I think we probably have some perspective, some interesting perspective on, or some insight into compared to, say, our students. Mm-hmm. So I'm curious if you have strategies that are sort of progressive in nature with regard to how you conduct research and instruction.
One of the things that has really been important for my lab is to discuss a very specific value statement and to think about all of the things that we do as a lab that have implications. Like, where do we publish our work? Mm-hmm. What are the values that those journals embody? Who are on those editorial boards? And just to have these conversations with the students about all the aspects of science that do have different biases. To start off with each student when they come in with a specific, like, here's the lab value statement and here's why we have these. Just to name it and to say out loud that we're not working as natural scientists in a vacuum here. Everything we do is impacted by society around us, and we have influence on all these processes. And then with that, it's then expected that everybody's thinking about these things on a regular basis and talking about them when they see them, and try to make space for folks to bring up problems when they see it, whether it be within our own lab or in the department, in the university. So that's one thing that I found to be helpful. And it also is front and center on our website, and we allow that lets students who are thinking about where they might want to go start thinking about whether the lab would be a good place to work. Yeah, those are not particularly innovative answers, but those are the things that I think get the conversations headed in the right direction, and then you can show your genuineness. But getting people who have felt excluded in the door is something that I think is really important.
So, because Erin is one of the smarter people I know, I'm basically trying to steal your ideas to help me in my future. Okay, so I do want to go back. So when did you switch at Bates to the natural sciences? What year were you
in a liberal arts college, you can take a while to figure things out. And so I ended up taking enough classes in poli-sci for a minor, but I didn't get it officially. I took enough Russian literature for a minor, philosophy. So I ended up being in environmental studies and a chemistry major. Mm-hmm. But I was really sparked by thinking about how very tiny things, very small changes can have global scale impacts. So that's what I was really drawn to. So I started doing work on metalloenzymes when I was in undergrad. And so these are enzymes that have a metal in them, and those metals are required for the activity that those enzymes do. So I got the opportunity to grow bacteria that were from hydrothermal vents, for instance, that had these enzymes. And I loved thinking about the activity of this invisible thing that I knew needed this tiny requirement, that activity had something to do with this amazing ecosystem at the bottom of the ocean. Then I got the opportunity to do some research as a research assistant in Woods Hole Oceanographic Institution, where I ended up doing my PhD. So I got the opportunity to go on a Southern Ocean research cruise, and the work was interesting enough, and I got to couple my love of being sort of in physically challenging situations. So the Antarctic ecosystems there's physical challenges associated with being there, and I liked that. I enjoyed doing hard work physically to do the interesting chemistry and the interesting biology. That was super satisfying to me. So I decided,
and we're talking about, you're on a ship, you're in the Southern Ocean. Yeah. You're talking about working on the back of the deck, freezing cold?
Working on the deck and going and taking ice core samples.
Okay, you're going on the ice. It's very, okay. So I need to know, where does this love of, were you always a runner when you were a kid? You were athletic?
I loved being outside, and I was reasonably athletic. I played softball, I played basketball, all that stuff. I spent tons of time hiking, and I eventually started trail running, and that became a passion for me, mostly when I was later in grad school and into my postdoc.
Trail running.
Yeah.
To me, there's a certain element of probably suffering in there. And like you talk about working, there's a certain element of suffering to working on the Antarctic ice sheets or icebergs, and working in the freezing cold temperatures. So is there an element of suffering in your writing, that side of your academic work as well? Are you like a punishing, stay up all night, or?
No. I don't really work very well like that. I like to, when I was in grad school, and then when I go to sea now, which is when I get to do my hands-on research, that I set up experiments that are absolutely punishing. But ultimately, they usually pay off. My students don't always love these ideas, but we have these big backpacks where we carry 20 liters of water in them, and we carry them between our incubator deck and where we do our water sampling, and so it's that part
on
of it
a rolling
is a little
ship.
And it's safe. They're in these backpacks.
I'm not saying it's not safe. I'm just saying it's hard.
It's hard, but we have our systems that make it possible, but they are challenging. And some of those experiments that have allowed me to make my bigger contributions to the field have been these things that, they're a bit punishing, and
you got to
not
wake up
everybody
every
wants to
2 hours to
or move way too much water around, or sample it in a way that's physically challenging. And but when it comes to the careful thinking and writing and the communicating, I don't do very well unless I'm reasonably well rested.
That research trip to the Southern Ocean was when you're an undergrad?
I had just finished undergrad, and I became a research assistant in a lab right out of undergrad.
Yeah. Okay, yeah. But you were looking at other labs, not just this one lab you were working in. Yeah.
I had trouble deciding. I knew I loved the work in the lab.
Trouble deciding.
Yeah, that's definitely a theme for me. But I mean, once I set myself on that path, it was easy to stick with it, and it was obviously the right choice for me. Yeah, all the things that I was deciding between, these were all labs who had really interesting new techniques to try to interrogate small scale processes, but always in the context of bigger biogeochemical cycles. Sure. So who's doing the nitrogen fixation at the bottom of the ocean and why, or how are microbes interacting with metals and mine tailings in ways that end up making them less toxic. So those were some of the kinds of questions I was interested in, but ultimately I decided the ocean is the best place to try to understand the impact of those very small requirements on large processes. So that's where I stayed for my PhD.
Scale up so quickly in the ocean, right?
Exactly.
And that type of thinking must have been relatively new. Like things like these large coordinated experiments like geotraces and things like that are relatively new ideas.
That's right. I mean, so one of the things that made this all tangible, when I was in undergrad and into grad school, this is when the ocean iron fertilization experiments has started being conducted, and those captivated my imagination and my sense of the idea that the research I wanted to do did have implications and ties to how we want to live as humans on this planet.
Right. So this was an experiment where they were putting iron dust in the ocean to try and make a big bloom of algae, phytoplankton, so that they would all die and sink, and that would be a way of removing carbon dioxide from the atmosphere.
Exactly. So in the early 90s, it became scientifically accepted that iron availability in the ocean limited the amount of growth of these phytoplankton in various regions. And so this sparked the idea that some folks followed up on, that if we dumped iron in the ocean, we would cause a big phytoplankton bloom. And perhaps that in the past had caused changes in glacial interglacial CO2 cycling and could be used as a tool in the future. Mm-hmm. And so those ideas were around, and I was interested in thinking about that from an ethical perspective on one hand, but I really wanted to know more about the chemical and the biological processes involved in what makes iron the limiting thing. What do the plankton do when they get it? What do they do when they don't have it? So all those kinds of questions I was really intrigued by. And then I started to realize that we knew a fair bit about iron, but we didn't know about some of these other elements or other molecules that plankton need in really small amounts. But if that really small amount isn't there, then large amounts of productivity just can't happen. Right. So, so that's what led me down the original research path there.
And not to jump too far ahead, but now we're sort of at a time where that's not only is it accepted, but like a tool that we're thinking about deploying. Is that fair to say?
Yeah, I mean, it keeps coming back, right? We think that we've shown that it doesn't actually have enough of an impact on CO2 removal from the atmosphere to the deep ocean. But people, I think, are so hungry for solutions now that, and this one seems like it has the potential to work. So even though we've come up with all these reasons why it probably isn't the best way to invest our resources monetarily or brain power wise on this iron fertilization as a solution, it does keep coming back.
You know, as someone who's thought about it for a long time, you for it or against it, mixed bag?
Well, so I don't think that it is the biggest bang for our buck in terms of where we can make a difference. And I don't think that the risks associated with it are worth it. And to me, one of the biggest risks is counting on it as a solution when it might really not do much to solve our problem here. So if we keep counting on this, we fail to act in other ways.
Is your lab looking into any sort of carbon mitigation?
We aren't working on it directly, and that's been a choice that I've made. I'm interested in understanding some of the possible consequences and trying to better predict the response of ocean ecosystems to some of these approaches and technologies. It feels like the Wild West right now, I guess I'll just say it that way. And so I don't have enough information about how the work that we would do would be used, or there's a lot of decisions that society needs to make before I'll be comfortable with deploying these technologies on a broad scale. I think it's important, and I'm not ignoring it. I'm paying close attention to it, but it feels like a bit of a scary space to me right now. Yeah.
Okay, so can you paint us a picture of your charming life in Cape Cod, Woods Hole, Massachusetts?
Yeah, I moved from basement apartment to basement apartment in Woods Hole. No, I loved Woods Hole. It's a very quiet place in the wintertime where, the community of students there is super tight knit because there's not really a whole lot else to do. And so that built a really nice environment for being a productive student where we would learn a ton from each other. So my work is super interdisciplinary now, and part of that is because I learned so much from my fellow students. So that was a super satisfying environment to scientifically grow up in.
Yes.
Yeah.
So could you just summarize what were the key findings from your PhD work? Yeah,
so vitamin B12, which is a cobalt-containing vitamin that humans need. Phytoplankton need it for the same reasons. So my major finding from my work was that the amount of B12 in the water in the Southern Ocean is sometimes not enough to support the growth of phytoplankton. So that can be a limiting nutrient down there. So this concept that a vitamin, which is something that is produced by a small subset of bacteria in the water, that could be what limits the growth of these phytoplankton, who are the major primary producers in ocean ecosystems. That was sort of a surprising concept to a lot of people. You don't need very many molecules of this thing in each individual cell to make things turn. But if you don't have enough of that.
More than zero.
More than zero. Yeah. I identified some proteins that are produced by some key plankton when they don't have enough B12. So this was sort of a sensor for whether they were stressed for having not enough B12. So we could go down to the ocean and measure the genes for producing that protein or that protein itself, and we can determine whether or not these plankton are likely to be experiencing some kind of consequence because they don't have enough B12 around.
When you were finishing up, the natural next move for you was to go to San Diego, California.
Yeah, in some ways it really was. I mean, to me it was because there was some research tools that I wanted to learn how to use that someone I had been collaborating with, he had been developing some tools in his lab that were really exciting and that I could see as the next big step for me in terms of being able to ask the questions that I wanted to ask about. And so I wrote a proposal to go work with Andy and to do some more Antarctic field work. And that got funded, and so I ended up at Scripps Oceanography and the Venter Institute in San Diego.
So if we could talk about just not really exactly the details of your work, but the bigger picture.
Yeah.
The idea that I personally find very overwhelming is that essentially you take a drop of water and there's everything in there.
Yep.
Millions or billions of things.
It depends on what you mean by things.
I mean, things that you could potentially measure, right? I mean, this is part of your work, right? You're talking about measuring essentially everything in there.
Yeah, I mean, so in my research, we measure metabolites, so compounds that cells produce, compounds that cells need, compounds that are generated from compounds cells make and spit out into the ocean.
Yeah.
Another line of research is measuring proteins in the ocean and trying to use those to understand what kinds of things cells need in the ocean and how those requirements relate to larger biogeochemical processes.
Right.
Yeah. So measuring proteins that cells make, measuring metabolites that cells make, how they transform. But to me, it's like we need to understand what those cells are making and doing, but we also need to understand the environmental context of that.
Right.
So what is it about the chemistry and the physics that led them to that? And what is it about the chemistry and the physics that's going to change, that's going to have important consequences for how those...
And so this is your project that you're a big part of, this BioGeoSCAPES. This is kind of the idea behind it.
And the idea is that, we measure all of the organisms in a drop of water and all the chemistry in a drop of water. Maybe it's going to be more like 50 liters of water at this stage, but we'll get there, maybe. So what is all the chemistry and the biology in any given parcel of water, and how does that relate to the major biogeochemical processes in the ocean, the future of our ability to extract food from the ocean, the future of the ocean's ability to serve the roles that it plays in global climate and carbon cycling right now. So the major innovation is that we would like to measure everything that's important about what microbial cells in the ocean are doing at the same time as we measure all of their chemical influences and impacts.
Yeah.
And so this is a big endeavor.
Yes.
So it requires an international program is the idea. So there have been programs that have done a lot of sequencing in the ocean before, and there have been programs that, like the GeoTraces program, that mapped the concentrations of metals all over the ocean, and this required technological innovations and then a global will to do it all together as researchers. So we'd like to bring those things together now and try to do this in a way that lets us understand what the future of microbes in the ocean is.
Right.
So there's still amazingly lots of questions left to be answered in that realm. And to really do it, we think we need this large-scale global map.
Yeah.
Yeah.
And there's, like you said, there's a big history of that being an important tool in oceanography, like in GeoTraces.
Yeah, exactly.
Once you plot these things on a map, they seem to make a lot more sense than they do on their own.
With the microbes, it's fun to think about like how important the temporal component of that is now too. So there's all these processes that happen, things that change over the course of a day, for instance, in biology. That so we can't just have a static map.
Yes.
And confronting that reality when we're designing this new program, it can't just look like GeoTraces. It can't just look like the World Ocean Circulation Experiment. Yeah. It can't look like that. So what are the key biological processes that we need to be able to capture? What are the right timescales? That's something that we're confronting right now.
There's also something like dimensionality that I find a little bit intimidating, that the space you're talking about is so multidimensional. It's not just one property in one place, but it's many linking properties that
You're totally right.
I can barely wrap my mind around.
That's why we think we need to have this concerted program for it, because there are interconnections between those timescales and variables that we know must be there, but we haven't been able to articulate, like what are the rules governing that relationship, for instance. And if we can't do that, then we can't incorporate our understanding of how these microbes are working into our predictive models of how the ocean's going to change, for instance.
So what's the roadmap for BioGeoSCAPES? Like what's it going to take to get this totally off the ground and running worldwide?
It's a huge question. But what it's most likely going to look like is there's a international science plan that gets identified and articulated, and then countries will take that back and figure out how we can best contribute.
Okay. Now, to tie it back a little bit, so when you finished your postdoc, you came here to Halifax.
Yeah.
Was this type of program something that you were visioning back then, or?
I originally thought I was going to focus more on some cellular details, where I wanted to figure out exactly what proteins were involved in managing the movement of vitamin B12 and a few others around a cell, and what are the real molecular details involved in that. And so I was excited about developing lab systems to try to interrogate that. But when I got here, I got more excited about getting back into the ocean in a bigger way. And so as time went on, more and more of my program was focused on getting samples from the ocean and understanding what was happening in the context of those samples, and then diving into that cellular detail from those ocean samples. And so it's been a bit different than I originally envisioned, but the questions remain the same, and the ultimate goal is the same.
A very interesting discussion. Thank you so much for coming in today. In our next episode, we'll talk to Dr. Michael Freund. He's a material scientist whose chemical research informs the design of sensors, electrical devices, and energy storage technologies. If you are enjoying Sciographies, hit subscribe and consider rating our show. I'm your host, David Barkley. Thanks for listening. Sciographies is brought to you by Dalhousie University's Faculty of Science and CKDU 88.1 FM in Halifax, Nova Scotia. Our producer is Nicole Killowy. You can learn more about Sciographies at dal.ca/Sciographies.